The overall goal of this procedure is to demonstrate the novelty of an anisotropic stretching platform that can be used to investigate single-cell responses to complex mechanical deformation and to quantify the mechanical properties of biological tissues. This is accomplished by first fabricating the flexible membrane and mounting this membrane on a set of clamps. The second step is to culture C2-C12 cells on the membrane overnight, and on the following day, mount the clamps on the stretching platform. Next, in a second set of experiments, thoracic aortic tissues are resected from a mouse model and cleaned from any connective tissues. Then, the aortic tissue is mounted on the stretching platform using a different set of clamps. Ultimately, the stretching platform is used to induce a deformation in cells along two orthogonal directions that can be quantified using an inverted microscope, or alternatively, a force transducer is integrated to the stretching device to measure tissue stiffness.
The main advantage of this technique over the existing techniques such as uniaxial or biaxial cell stretching is that it allows us to control independently the deformation in two orthogonal directions in a transparent membrane on which cells are attached, and therefore allowing for live cell imaging microscopy. Also, a careful design of a different set of clamps with the integration of a force transducer on the platform allows us to perform tensile testing on biological tissue for the quantification of their mechanical properties.